So thanks, John. That was really kind. And thanks everybody for coming. I didn't expect such a big crowd. I'm thrilled to see so many people here. So I'm going to try to address this question here, which is how the brains of people with autism differ from typical brains, which seems to me a pretty central question we're going to need to answer if we're going to understand autism. I'll say right now, I don't think I'm going to give you a very satisfying answer, but I'll tell you about my efforts to date in this venue. So one of the things we know about autism that comes from the very definition is that at the very core, it's a deficit in social cognition. There are all these other phenomena that go along with autism, possible attentional effects, motor effects, repetitive behaviors and such, but I think people generally agree that social deficits are really at the core of the phenomenon. So I'm going to focus today on looking at the social brain, that is parts of the human brain that are involved in different aspects of social cognition, since that seems most likely to tell us something about what's going on with autism. Okay, so what is a social brain? Here's a cartoon picture of a brain with a bunch of regions which have been implicated in different aspects of social function. And the first thing you notice is it's a lot of the cortex. There's a lot of, just in terms of area of the brain, a lot of it is involved in carrying out different kinds of social functions. Another thing that you see even in this little cartoon here is that it's not all homogeneous, as John alluded to. There are different bits in there, many of which are functionally very specific. I'll talk about some of those today. One that we started off working on about 15 years ago was the fusiform face area, which is actually not right there. It's actually underneath where you can't see it. But that region is very selectively involved in processing faces. A region that is primarily the work of Rebecca Sachs and her students, this yellow one here, the TPJ, is quite selectively involved in thinking about what other people are thinking. That's kind of a miracle. It's kind of wild, but it's true. She's shown that very robustly. And these red regions here have been known to be involved in processing language for a long time. And Ev Fedorenko, working with me over the last 5 years or so, has collected, I think, really impressive data showing that those language regions are actually very specifically involved in processing language. They don't also do double duty for, doing mental arithmetic or working memory or cognitive control or any of these other functions. Okay, so these regions are very specific. Each of these here has been replicated in many different labs. They are pretty well characterized in typical subjects. That is, for each of these regions, we know a lot about how they behave and what different things they respond to. And each of these regions is present in pretty much every typical subject, right? So they're just part of the basic architecture of the human brain, and I would say of the human mind as well, which is really why we care about the brain. And so it seems to me this is a good place to start when trying to understand autism. So just to hit that meta point for a little bit longer, this suggests a research program in autism where we can start by saying, well, first of all, do people with autism have all this basic architecture? Do they have all that machinery? And is it of a similar size and selectivity and location in the brain? But then we can also ask whether those regions behave similarly. And behave is an intentionally vague word that can mean all kinds of different things. Do they have similar connectivity? Do they interact similarly with other regions of the brain? Do they contain similar kinds of information in people with autism as with typical people? So that's roughly the research program I've been trying over the last few years. I can't say that it's been massively successful, but I'll tell you where we've gotten with this enterprise. Right, a little more meta stuff first. The reason that it seems to me a good idea to start with these things rather than other approaches where you might, for example, think up some mental process that seems particularly implicated in autism, devise a new innovative task that you think taps directly into that mental process, scan a bunch of typical subjects and a bunch of people with autism, and look for a difference in the activations. That's a perfectly respectable approach. Lots of people do that. But I think this approach has a number of advantages over that, which I'll mention here. First of all, each of these regions here is extremely robust in typical subjects. So we have kind of a solid foundation to start with. That means that it's easier to detect a difference in autism. We know what those things look like in typical subjects. So if they're going to be different in people with autism, we have more of a basis for detecting it. Second, each of these things can be found in pretty much every typical individual typical subject. And that's useful because we could, in principle, detect heterogeneity in a group of people with autism. Heterogeneity being one of the key methodological challenges in studying autism. That's not one thing, it's a big smear of different things. Third, because these things are pretty well functionally characterized in typical subjects, that means we have a leg up in understanding what differences in these regions might mean, right? If we find that one of these little colored bits in here is absent or smaller or different in people with autism, because we know a bunch about that region in typical subjects, we can make more of that result. And the final reason I think this is a good idea is that, in a sense, by studying these regions, there's a whole set of procedures for finding them and identifying them and measuring the magnitude of their response. You're essentially tying your hands in a good way. I think a lot of neuroimaging work suffers from excessive degrees of freedom. That means you have this huge data set with enormous numbers of dimensions, and you can fish through it every which way. You will find something in there. You are guaranteed to find something in there. But a lot of the somethings you will find will be spurious. And in contrast, if you start by tying your hands and really restricting your hypothesis space, you're less likely to discover something spurious. Okay, enough meta stuff. Degrees of freedom. Okay. This enterprise is in very early days, and most of the data I'll show is pretty preliminary, but here's what I'm going to try to talk about. So first of all, we're going to look at those face processing regions and ask if they're different in people with autism. Second, we'll look at those language processing regions and ask whether they look different in people with autism. And third, I'm going to briefly mention some data that I have nothing to do with. I just think these data are really cool. This is work by Rebecca Sachs and Jory Koster-Hale. And finally, I'm going to talk about, do I have a pointer here? Is this a physical? Oh, wow, cool. I feel so powerful. I lost my train of thought. Yes, this region up here. This superior temporal sulcus, which has all kinds of interesting stuff in it, and where I think it's less understood in typical subjects. So I'll talk about some of the typical data about the functional organization of that region. Okay, that's the agenda. So let's start with this face stuff. So we started off doing the very obvious thing that other people have done before, which is simply to scan subjects with functional MRI while they're looking at images of faces, scenes, objects, or bodies. In this case, we're showing them movie clips, little 3-second movie clips that we shot. We shot movies because we thought they'd be more engaging for scanning kids. Actually, most of the data, I think all the data I'm going to show are from adults. It's really hard to scan kids. You get a lot of artifacts. I'm not talking about those data. I'm talking about adult data today. And so the way you find those face processing regions in pretty much any subject is to scan them in a paradigm like this, and then just do a contrast over the whole brain and say, show me the regions that respond significantly more when subjects are looking at faces than objects. Okay, when you do that, you get a bunch of regions, like these blue ones here. This is a bottom surface of the brain. So if you looked at my brain this way, this is the temporal lobe here, back of the head, front of the head. Everybody oriented? It's inflated so you can see the whole cortical surface. The dark gray bits are the bits that were inside a sulcus, a fold that got inflated so you could see it. So this big blue patch there is the fusiform face area. And this is a face selective region in the superior temporal sulcus. This is in one subject here, just to get us all oriented. Okay, and other regions that are shown here. This is a region that responds selectively to scenes, not, I would consider, part of the social brain. And the green bit responds more strongly when you look at images of bodies and body parts than objects. Okay, so, oh, right, so this is just a lame excuse to show you a fun video that I made for that TED Talk. Okay, so this is my brain. And I scanned the hell out of myself before my TED Talk so we could find every functionally specific region that I or anybody else has worked on. And so the pink stuff are my language regions. Oh, now I'm doing a little half pipe flip here. The red ones the red ones on the bottom are my fusiform face area. The white ones I'll talk about briefly. Those ones are very general. They respond to pretty much any kind of mental effort. They're sort of the opposite of all the other regions that respond for very specific kinds of stimuli and specific kinds of mental operations. Okay. Oops, no, we did that enough already. Okay, here we go. Okay, so now what do these things do, focusing on that fusiform face area and the STS face region? Well, the usual story that you find, which I think is probably approximately right, is that this region back here is more involved in just perceptually representing face identity or face structure, really the sort of basic perceptual aspects of processing the shape of a face. Whereas these regions up here are more involved in kind of higher level kind of social stuff, like perceiving direction of eye gaze, perceiving emotion, and perhaps other things. So that's the usual story, and I think that's approximately right. Here's some data that David Pitcher and I collected a few years ago, showing the selectivity of this region. So when I say these things respond selectively, here's an example of what I mean. This is now the magnitude of response. You identify that region, and then in a separate bunch of data, you measure the magnitude of response in that region to a bunch of different conditions. And you can see it responds a whole bunch more to faces than anything else, and similarly so to the superior temporal sulcus face region. Okay, so if everybody oriented, that's just showing you what we sort of know already. But then David Pitcher's experiment found something else pretty cool. So what we'd had was we just made these movies because we were scanning kids, and we thought, well, that's nice. They're more fun to look at. And then everybody found them more fun to look at, so we just used them as our standard way to identify these regions. But early on, we wanted to make sure that you pick out the same regions with movies that we'd been picking out previously with stills. So we took the movie clips that were just little 3-second clips, and we took a 1 second still, 3 1-second stills from each 3-second clip, and presented those in the static condition here. And as you can see, the fusiform face area doesn't give a damn. Like movies, stills, it's all the same. It doesn't care. But so that's percent signal change. So 0 is the response in that part of the brain when you're staring at a dot. Okay, so you can't turn the brain off, but for visually responsive regions, the most you can turn it off is have people fixate on a dot, because there's no eye movements, there's no stuff to look at. That's the best we can do. So it's what percent increase does the MRI signal go up from there for each of these conditions? Yeah, and we get really excited by 0.5. Well, so that's huge, right? But keep in mind that that's a percent increase in the MR signal. The MR signal is a very indirect measure of neural activity. And so it does not mean that it's a 0.5% or 1% increase in neural activity. What percent increase in neural activity it is, we don't know from this. It's just an abstract indicator that neural responses went up. Okay, so the FFA doesn't care about moving versus static things, but this region in the STS cares deeply. Okay, so we don't know what that means, but all of the hypotheses I can think of about why that region would care so much about face movies are interesting. So I'm intrigued by that. I can't tell you why it does that, but I think that's pretty cool. Okay. So now we can ask, are these regions affected in autism? So we just do the same thing. We scan adults with autism who've been properly ADOSed and all of that on this very same paradigm. Oh yes, background. Yeah, lots of people have asked this question before. So there's a paper by Bob Schultz, an early one, where he said, yes, there's a big difference. People with autism basically process faces as if they were objects. Okay, that was a very compelling story. It was kind of a meme that took off, and there's no stopping it. There was another paper that said basically the same thing, and then shortly thereafter, people started repeating these experiments. There's a very nice study by Hajikani that found basically the FFA response is pretty much the same in people with autism and typical people. But that study isn't cited as much. So anyway, it's been done before, but we thought we'd give it another look. Okay, so we scanned. This is done with Alex Kell, shown here. We're still collecting subjects here. I think given the heterogeneity, you just need a lot of subjects. They're hard to get. So we're at 19 adults with autism right now and 31 typical subjects. And basically, here's what we find. This is now the volume of the fusiform face area. So you count up the number of voxels in there that reach significance at some fixed threshold. There are other measures that show the same thing, but basically, how big is that region? So basically, there's no difference in our typical group and our autism group in the FFA. They all have FFAs just fine, and the FFAs aren't smaller. So the basic region is there. Okay. And as a bunch of control regions, the body selective region and a bunch of scene selective regions also are very similar in the adults with autism and the typical subjects. But look what happens with that STS region up there. It's a whole lot smaller, right? So that's this bar right here. So that region, a lot of subjects, you don't see anything at all, any activation up there at all for those face movies compared to object movies. And the ones that have it have much less activation. It's much smaller. So the fusiform face areas doesn't seem to be affected, at least in these data. And actually, I'm not surprised by that, not only because of the prior literature, but because I think basic face discrimination is probably not strongly affected in autism. And we have a bunch of behavioral studies where we've looked at that and found that people with autism are, actually, this was kids with autism, are very similar in their ability to discriminate similar faces from one from another as our typical subjects. Yeah? Yeah. Yes, these regions are bigger in the right hemisphere than the left, and there's no difference in the degree of lateralization of these regions between the autism and typical subjects. But I'll get to lateralization when we get to language, where we do see differences in autism. Okay, so I'm not surprised that the FFA doesn't seem to be different. I think it's doing these basic perceptual discrimination of face processes. But I think it's cool that that this STS region is different. And that really underlines the need to understand what that region is all about in typical subjects. And I'll get back to that in the fourth part of my talk. Okay, so on this first part, are these basic components there? Yes, FFA and these other regions are present and look normal just in terms of their volume, but the face region in the STS is compromised, and we'll return to that. Okay, let's talk about these language regions. This is work with Ev Fedorenko. Actually, that understates the case. This work kind of is Ev Fedorenko. I'm just sort of, I get to follow along and see the cool things she does. So first, a little background. About 5 years ago, Ev started by doing functional MRI studies in typical subjects looking at language parts of the brain. And of course, language parts of the brain have been known for 200 years, since Broca and Wernicke, so there's no surprise about where they are. But within the functional imaging literature, what she did that most people don't do is look at individual subjects and identify regions in each subject individually. Because we think that the location of these regions is variable across subjects, and by mushing across subjects, you lose a lot of the precision of the functional specificity that you see in individuals. So she started off by contrasting the response when people read sentences versus when they read strings of pronounceable non-words. Okay, in both cases, you have to remember the string, whether it's a sentence or a list of non-words, because you get a probe item at the end, and you have to say whether that item was in the sentence or the string. Okay, so actually that task is a lot harder in the non-word task than the sentence task. We wanted to kind of push mental effort in the opposite direction of sentence processing so that we didn't have an effort confound with the sentence responses. And you see here in two typical subjects, each an individual subject, you see nice, robust activations in all the places you'd expect. Okay, so no surprise there. To be able to identify those regions in each subject, Ev devised a nice way to kind of look across subjects at the broader region where these activations land. That's those red regions. And here's a few individual subjects shown in here. And so what she's done is devised a way to basically and each individual subject's activation with those broader parcels to define in each subject 8 different regions in the left hemisphere. Okay, that's just a way to kind of say what's what across subjects. Okay, and various reality checks. These regions respond to sentences whether you read them or hear them, as any proper language region should. This is not a region involved in visual perception or auditory perception. It's involved in high-level linguistic structure, the syntax and semantics of the structure. And then one of the next things Ev did was to say, okay, now that we can find these regions in each subject individually, let's ask this classic question of whether language is segregated in the brain from the rest of cognition. So do these regions that respond more when you read or hear a sentence, are they also more engaged when you do all of these other mental tasks that have been argued to overlap in the brain with language processing? So mental arithmetic, lots of working memory tasks, cognitive control tasks, all these things, music, all these things that people have argued overlap in the brain with language processing. And no, they don't overlap. When you define those regions in each subject individually, you find, for example, this is essentially Wernicke's area and essentially Broca's area, a high response to sentences compared to non-words, and pretty much no response to any of these other conditions. And it's not that these other tasks don't activate the brain. They activate nearby regions very robustly, just not these regions. Okay? So that's cool and important because it means we can target those regions, and we're really looking at something about language. They're really a marker for something about language, not about any old aspect of high-level cognition. Okay? Okay. So that means we can now ask in autism whether these regions are present, whether they're of similar size, whether they show a similar response magnitude, and whether they're lateralized to a similar degree. Okay? Okay, so to do that, this is also collaboration with Alex Kell. We started scanning adults with autism, high-functioning adults with autism, on very much the same task. Here's two of them up there. If you just eyeball it, you can see it looks really similar. It is pretty similar. So then we figured we should get a little more quantitative. So we're still scanning subjects, but the data I'm about to show you has now 21 adults with autism and a very large pool of typical subjects as a baseline, because Ev uses this localizer in all of her experiments, so she's got nice baseline measures. And so first of all, do they have similar, are these regions of similar size? So what I'm going to show you is 8 of those language regions that I showed you before, identified with that little parcel method. I'm going to show you the volume of each language region in typical subjects and autistic subjects. And you see here in the left hemisphere that they're pretty similar, but if anything, the people with autism have larger language regions than typical subjects do. And same deal in the right hemisphere. In fact, even more so. So basically, if you look in here in these temporal regions, there's substantially larger language activations in the people with autism than the typical subjects. So basically, these regions are larger in ASD. That increase in size is greater in the temporal lobe than the frontal regions. And it's more so in the right hemisphere, leading to what turns out to be a pretty sizable difference in lateralization, where this is now a measure of how left-sided, how lopsided on the left are those activations. And what you see is that they are more lateralized in typical subjects than people with autism. So people with autism have bigger language regions, and there's more on the right. Okay. Okay. So, and this is your question before, does this happen all over the brain in autism? No, it doesn't. Here's the fusiform face area. There's no significant difference in lateralization, and a bunch of other visual regions that show no difference in lateralization. So it seems to be something about those language regions in particular. Okay. All right. Okay, now, here's this language network that I've been talking about, this set of regions that respond selectively to language. I mentioned before that these kind of astonishingly nothing responses to all of these other tasks don't result from a total absence of response to those tasks. In fact, oops, hit the wrong button here. In fact, there's a whole other set of regions that have been called the multiple demand system by John Duncan and other people. What he means by that is this other set of regions those are those white ones I showed you on my brain. They're the ones that are engage, apparently, when you do anything difficult at all. They don't care if you're doing that difficult thing on a sentence or a face or an equation or what it is. These regions get engaged. And so just to show you what that looks like, here is the response in a couple of those multiple demand regions defined in each subject individually across this suite of tasks. And the solid bars here are a difficult condition versus an easy condition. This is spatial working memory, difficult versus easy. This is verbal working memory, difficult versus easy. Basically, every contrast of difficult versus easy, no matter what it is, engages that same region. So these regions are just completely domain general. They'll kind of do anything. Whenever you're doing something difficult, they step up and engage. Okay, and there's a whole interesting literature on these regions. As one little sidebar, John Duncan and his co-workers have shown with lesion studies that these regions are related to IQ, in the sense that if they studied a pool of, I think, 80 patients who had brain damage, and they quantified the volume of brain damage in these regions versus the volume of brain damage outside the brain. And they found that IQ goes down linearly with loss of those regions and is unaffected by loss in the other regions. Okay, so if you lose the other regions, you lose face recognition or language function, or you become deaf or paralyzed, your IQ doesn't go down. But if you lose these regions, your IQ goes down. Okay, so that's a very interesting system. It's different than the language system. And now we can ask, what is the relationship between these two systems? Okay, so there are lots of ways of doing that, none of them perfect in humans. But I'll show you one that I think is pretty cool. This is work with Ev Fedorenko and Idan Blank, who's a grad student in the lab. And these guys decided to use a method that's been around for over a decade that lots of people use. And we only weren't doing it in my lab because I was just kind of a curmudgeon and I didn't really understand this method and I didn't want to use it because I felt we didn't understand it. But it turns out it's pretty interesting. Okay, so in the method, we define each of these regions. And then you scan subjects while they're just lying in the scanner doing nothing in particular, at rest, whatever that is. They're just lying there, okay? And you extract the time course from each of these regions identified functionally, right? And then what you can do is ask whether those time courses at rest are correlated across two different language regions within a subject, or across two different parts of this multiple demand system, or between language regions in multiple demand regions. Okay, so the reason I hadn't messed with this method is nobody knows exactly what these correlations mean. They are not a pure measure of direct structural connectivity. There are parts of the brain that produce strong correlations with this method that we know are not directly connected, which is what made me nervous. Nonetheless, I think when you see the data, you'll see there's real structure in these correlations, whatever it is they mean. Okay, so I'm going to show you a correlation matrix. So we take all of those regions of interest, all of the language ones up here, the multiple demand ones here, and we just look at the strength of the correlation at rest across those regions. And what you see is a big square for all the language regions. Basically, every language region is correlated at rest with every other language region, particularly in the uppermost square. That's the left hemisphere language regions. Those are probably the real deal. And all of the multiple demand regions are correlated with other multiple demand regions, but there are not correlations between the two systems. So here are these big brain systems that span, from the temporal lobe up to the frontal lobe for the language system and the parietal lobe up to the frontal lobe for the multiple demand system across big expanses. They're all pretty strongly correlated despite those big distances in the brain at rest, but they're not correlated between. So here's the mean correlation between any two language systems, between any two multiple demand systems, and between a language and a multiple demand system. So that's pretty neat. As I say, we don't know exactly what it means, but there's real structure here. It means something. So then we can ask whether you see this in people with autism. Answer, it looks exactly the same in high-functioning people with autism. So I'm sorry about the long string of null to small results, but that's just what it is. Like this, I think it's pretty interesting. It says that not only are the basic language regions present, and in fact even larger, they're showing the same patterns of correlations with other systems that we see in typical subjects. Okay. Okay, so to summarize all of this, and as I say, we're still collecting subjects. I think given the heterogeneity, 20 subjects isn't enough. I think 30 is a bare minimum. We're having a tough time recruiting, but we just hired a new recruiter, so I hope we'll wrap these studies up soon. Anyway, yes, these basic systems are present with more or less the same topography and high-functioning people with autism. They are even larger in autism. They're more bilateral, less lateralized in autism. And they seem to show similar interactions with other regions of the brain. So frankly, I don't know what all these differences mean. What does a larger range of activation, a larger region of activation mean? Who the hell knows, right? But there are some precedents for the idea that a larger activation reflects a less efficient neural activity. That's been shown before in motor cortex. And actually, Ev Fedorenko has also seen this. I don't know if you guys saw around a year or two ago, there was this high school kid who knows, I forget how many languages, 30, 40, some insane number of languages. The guy's a real prodigy polyglot. Well, she brought him up to Boston and scanned him and looked at his language activations, and they are tiny. So who knows? Maybe when your system becomes more efficient, it produces a smaller activation. And as far as the reduced lateralization in autism, I think that's interesting. It's certainly been seen before, although not with this individual subject method we're using here. But it's not very specific. You see this also in schizophrenia, specific language disorder, and epilepsy and other disorders. Yeah. Yes, we balance for that in these comparisons. Yeah. Yeah. And it doesn't make a difference much as Jim wants it to. I just showed him all my non-differences in the typical subject. So he's interested in the idea that what happens in autism is that the reason you get an uneven male-female ratio in autism is in part that girls start with an edge in social cognition, and that you would see these differences even in a male versus female contrast in the typical population. And I resisted his exhortations to look at this for a long time because I'm allergic to sex differences in the brain. I don't trust the public with sex differences in the brain. The whole thing makes me nervous. But at his urging, I looked, and we don't see any significant sex differences in any of the measures I'm going to tell you about here that show a difference between people with autism and typical subjects. There are some trends that he's excited about, but they're not even close to significant. Okay. So given that we don't yet know what this means, what do we do with this? Well, I think the next step is to try to figure out what the lateralization means and what the size of activation means. And I think one way to do that is to go back to typical subjects and look for correlations with language abilities and other things between these measures. So we're now engaging in that enterprise. And I think the other big thing that will help here is to have more sensitive measures of language processing instead of these really crude things like, is the system there? Well, yeah, the system's there, but so what? You want to know not just that it's there and how big is it, but what information is represented in each of those regions. And so there's some nice methods to do that. And actually, this is in part why I brought up the this work by Jory Koster-Hale and Rebecca Sachs, even though it's not my work, I think it's a really beautiful illustration of how we can use subtler measures with functional MRI to find real differences between people with autism and people without. Okay, so just to give you the background, I mentioned this briefly before, but starting around 10 years ago, Rebecca Sachs has been studying a bunch of brain regions, but especially this one shown very schematically here, the right TPJ. And that region responds very strongly when you do any task that requires you to think about what another person is thinking. And what's astonishing about this region and about Rebecca's data is the specificity of that response. So she's tested a large number of other conditions. This region is not engaged when you do logically isomorphic tasks about non-mental representations, right? So in the in the mental representation task that activates it, you read stories about what another person knows or thinks, and you have to reason based on their knowledge what they'll do. In the control conditions, she's got people reasoning about a photograph or a map, a physical representation that also can differ from reality. So it's logically the same kind of structure. It's just not a mental representation. It does not activate this region at all. She's also shown that other kinds of social tasks, like reasoning about what a person looks like, even very vivid, engaging, unusual-looking people and unusual cultural practices, this region is not interested in that at all. Weird cultural practices, it doesn't engage. Even reasoning about a person's physical, bodily sensations like thirst or pain or hunger produces no activation here. This region is only activated when you think about the content of their thoughts. Okay? So that's really extraordinarily specific. And that makes it a really prime target for work in autism, because this is really at the core of the deficit in autism, is reasoning about other people's thoughts. Okay, so we can ask, does this region exist in high-functioning people with autism? Now, as a little sidebar, the way you find that region is to scan people while they're doing a task that requires them to reason about other people's thoughts. So if you scan somebody who can't do that task, that doesn't make any sense. So you'd only run these experiments on people, high-functioning adults with autism, who can now do that task. They probably developed it later. They may be a little slower at it, but they can basically do the task. And when you scan them doing that task, this is work with Rebecca's student, Nick Dufour, because she also uses this contrast of reasoning about other people's thoughts versus reasoning about physical representations to localize that region. She has lots and lots of typical subjects, 460 in this study. And when she compares those with 30 adults with autism, she finds no group difference at all in any measure of that region. It is there, it's in the same place, it's in the same volume, there are many different measures of the nature of that activation. It's just indistinguishable in high-functioning people with autism versus typical subjects. So on the one hand, it's like, what's up with that? That's weird. And on the other hand, theory of mind, that is thinking about other people's thoughts. Okay? And on the other hand, well, this is a crude measure. Maybe it's not that surprising. The basic system is there. Here it is. It turns on in the same tasks. But does it work the same way? So to look at this, Rebecca and Jory Koster-Hale, shown here, asked a deeper question, and that is, what information is represented in there? So what they did is they took the following scenario. They had shown that in typical subjects, when you scan them, doing tasks that require them to understand the distinction between accidental harm versus intentional harm, not only is this region engaged, but if you look at the pattern of response across that region, you can tell if the person is reading a story about accidental harm or intentional harm. So let me give you one of their examples from their study to show what I mean. So in this paradigm, the subjects are lying in the scanner, reading stories like this. Your family is over for dinner. You wish to show off your culinary skills. For one of the dishes, adding peanuts will really bring out the flavor. Okay, you grind up some peanuts, add them to the dish, and serve them to everyone. Your cousin, one of your dinner guests, is severely allergic to peanuts. The plot thickens. Okay, now there's two conditions, right? In the accidental harm condition, you then read, you had absolutely no idea about your cousin's allergy when you added the peanuts. Okay, in a different bunch of trials, you would read, in the intentional harm condition, you read, you knew about your cousin's peanut allergy when you added the peanuts to the dish. Okay, so the TPJ, that region of the brain, you can tell by looking at the pattern of response in that region whether you're reading a story about accidental harm or intentional harm. So that's pretty fascinating. Further, in some other studies, Rebecca and her student Leanne Young showed that if you zap that region with transcranial magnetic stimulation, which messes it up transiently, you reduce people's appreciation of this distinction when they make moral decisions. Okay, so that region is causally involved in representing this distinction.
What's the representation look like if it's intentional versus
Oh, it's nothing that you can make any sense of. It's just a pattern of voxels that has a response profile like this for intentional and like that for accidental, you know.
Does it look different?
It doesn't, if you look at the data and you give it to a machine learning system and say, look at those data and tell me which is which, it can perform well above chance. But there's nothing you can make sense of, like this voxel's a little higher here and that one's, it's just some pattern that there's no direct way to interpret, right? Okay, so that information is in there in typical subjects. All of this is preamble to say, and in typical subjects, the magnitude of response, not the magnitude of response, the amount of that information present in that region is also predictive in typical subjects of the degree to which they respect the distinction between accidental and intentional harm in making moral decisions. Okay, so it predicts individual behavior in typical subjects. Okay, so what do you see in people with autism? Oops, wrong way. This information is absent in the TBJ of people with autism. Okay, so that's pretty cool. It says that the basic machinery is there, the basic system is there in people with autism, but it works differently and doesn't represent the same information. Okay. That's right, and people with autism pay less attention to that distinction in their moral decisions as well. Yep, and you see it in this part of the brain. Okay, so all of that to say, here's a case, I just said this, the region is there, but it's functioning differently and not representing this information that's represented in typical subjects. Okay, so that's pretty cool. I forget what my segue is. I don't really have a segue here. On to the next thing. Okay. That's the temporoparietal junction, that little yellow blob, which is my schematic representation of that region that's engaged in thinking about other people's thoughts. Okay, so last section, this whole region that I've stuck the white oval over there has been implicated by many people for a long time in different aspects of high-level social perception. Okay, so I think I put, so here are two on the left. This is a review article from almost 15 years ago from Truett Allison and Greg McCarthy. And even back then, there were loads of studies that had already shown activations in that region. So this is the superior temporal sulcus, this very long sulcus that goes way along here. And all these studies had already shown strong responses with functional MRI in that region when people look at different kinds of social perceptual stimuli or hear them in some cases. And in a review article from a bunch of years ago, Hine and Knight made an updated figure like that, and they looked at these a bunch of different functions from different studies and stuck a dot up there for each study, and they said, look, this stuff is all interleaved. You have motion processing, speech processing, theory of mind, audiovisual integration, and face processing, all in that same area. Therefore, they say, this argues against a strict functional subdivision of the superior temporal sulcus. Well, that doesn't follow from these data. It doesn't follow from these data because those points up there are from different studies and different people. Further, every one of those points is just the peak from some activation that's a group analysis that's a blurring of each of the individual subjects. So we really have no idea from these kind of representations of the literature whether in fact there's a strict segregation up there or not. The only way to find out is to scan all of these tasks in an individual subject and ask whether in that subject these things are segregated or not. As soon as you go pooling across subjects, you're going to blur things anyway. You're not going to be able to ask this question of specificity. Okay? Okay. So this is what Ben Dean and Rebecca Sachs and I have been up to for the last few years. That's Ben up there as a grad student in our program. And when I first said to Rebecca, we have to tackle this, she said, don't you remember I tried answering that question when I was in graduate school, and I didn't. But anyway, we're trying again. I'm not sure we're doing much better, but anyway, we're doing a little better. Okay, so first thing is basically the simple idea of let's take all of these previously reported tasks that activate that general region, let's run them within subjects, okay? And then so the first thing I'm going to show you is, so this is an anatomical mask here following the sphirotemporal sulcus. So we look at that whole region, and then we look at the pattern of response across that region for each of these contrasts. So these are two different theory of mind contrasts, just two different paradigms. At Fedorenko's language contrast, two different ways to look at voice processing. So Pascal Bélin has been doing a whole series of studies arguing that you get responses up in the sphirotemporal sulcus when you listen to human voices. He argues that that response is not about speech per se, because the same region responds when you hear non-speech vocalizations, like crying and sighing and laughing. And then we have face responses up there, our face movies. Okay, so what we can do then is ask how similar the pattern of responses across the whole superior temporal sulcus for these different contrasts, right? So if it was all one multifunctional region that was equally activated by all of these things, we should have a strong and even correlation across all of them. But instead, what you see is, the data are split in half before you do it. You first see nice replicability of the pattern of response across that region between the two halves of each task. That's what's shown on the diagonal, those correlations. And you also see nice kind of replication. So here's two different sets of theory of mind tasks, and there's a correlation in the pattern of response from theory of mind contrast 1 to theory of mind contrast 2, as there should be, but it's nice when things work. Same deal for the two different voice contrasts, but basically all of these other zones are pretty blue. They're not correlated. That tells you it's different. There's a different pattern of response across that region for these different contrasts. In other words, they're not all one big multifunctional thing. There's some structure in here. Okay? Okay, so let's zoom in on those face activations. Because recall that I showed you way back at the beginning of the talk, here's a case where we really see what looks like a big difference in autism. Although the fusiform face area response is the same volume in people with autism and typical subjects, this region in the sphirotemporal sulcus is really quite a bit smaller in people with autism. So that seems like a promising zone to dig in. And so what we want to know first is, what does this region do? So the first clue we already have is that it cares a lot about face motion, right? So I don't know where to go with that, but I think it's very very compelling. There's something you get from a 3-second movie clip of a face that turns on that region much more strongly than 3 consecutive stills in the same order from the same movie clip. I mean, they're really similar, and yet not to this part of the brain. So there's something that video supports perceptually that the stills don't. Okay, but what about other functions that have been ascribed to this region? Okay, so Rebecca and Ben and I looked at that face responsive region, and we measured its response to all of these other tasks here. So this is a smorgasbord of all the contrasts that people have used to show activations in the sphirotemporal sulcus. And first, what you see in that right face region is a higher response to faces than objects. Okay, that's how the region was defined, so it better do this. I mean, this is left out data, so it's a replication, but still, that's expected. Interestingly, this is the theory of mind contrast, thinking about person's beliefs versus thinking about physical representations. No difference at all in the theory of mind contrast that activates the TPJ very strongly. A little bit of a higher response to biological motion. I'll show you some of these later if I have time. These are moving dots that look like a person walking, but not as strong as to faces. Okay, so that's all reasonable and more or less as expected. But here's a surprise. It responds just as much to human voices as it does to faces. Now, this is embarrassing and revealing and kind of fun all at once. Lots of people, including me, have published papers talking about this region as the STS face region, because we had tested a bunch of conditions and it responded a whole lot more to faces than anything else. And we didn't really know what it did, but it didn't really occur to us that it would do anything as scandalous as this. Well, it turns out, it's not a face region at all. It responds at least as much to voices, okay? And you can see also over here, these conditions are all auditory conditions. So this is listening to sentences. This is another theory of mind contrast. But it's also voices, and that same region produces a strong response to all of the conditions that have voices in them, whether it's speech or non-speech vocalizations. So that's interesting and scandalous. So this is frustrating because we still haven't answered this. So the first two hypotheses we came up with are, okay, what is it that would be engaged when you look at moving faces and when you hear vocalizations? Well, maybe some kind of audio-visual integration that we do, in fact, more with speech sounds and non-speech sounds, but maybe that's close enough. And the other, oops, what am I doing here? The other thought is maybe this region is responding to communicative intent in the stimulus, right? So when you hear vocalizations, whether speech or non-speech sounds, usually somebody's trying to communicate with you. And if you look at faces, very often they're trying to communicate with you with their face, whether they're speaking or not. And so that seemed like a good hypothesis. Unfortunately, we've refuted both of these in an experiment that I'll tell you about offline if you ask me, but it's a little complicated, so I axed it from the talk. There wasn't room. But just to give you a few examples, we filmed people making gestures with their hands, some of them extremely communicative, stuff like this or this, all kinds of gestures. You look at these hands, they're really lovely videos. They're extremely communicative. This region doesn't respond to those practically at all. It also doesn't respond differentially to face motions that are communicative, like a video of a person talking versus a video of a person chewing. It likes those both. What? So I'm kind of stumped with this. I don't know what the damn thing is doing, but it's doing something interesting. It's not. It's implicated in autism. It does not respond to all of these conditions, and we will nail it. We just haven't yet. So instead of pursuing that frustrating and ultimately still hanging an unresolved question, I'm going to change the topic slightly and tell you about a nearby region. In fact, it's right next door to this region, sometimes slightly overlapping with this region, that does something we have a little more of a handle on. So this is work by Cami Coldewine, shown here. And she asks the question of whether we have specialized machinery in the brain for perceiving social interactions between two people. Okay, that's something we do a lot. We care about it. We're really good at it. So she started by using, okay, so here are the point light walkers. This is from another study by Manara. So if you just look at this, you'll see a very rudimentary social interaction. This person says stand, the other person stands. Okay, it's not deep, it's not amazing, but you can see that they're communicating with each other. In contrast, here's a video we made where we took that top case and we just flipped it like this so they're facing away, and we stuck a line down the middle so that they're perceptually pretty similar, but it's not as clearly communicative, okay, because they're facing away and there's that wall in the way. I find this only partly effective. The timing is such that I see an interaction in there anyway. I don't know. I think it's not perfect. So because I felt we weren't able to destroy the percept of an interaction here, we went to a slightly more extreme maneuver where we took just cases of people involved in clearly independent activities like this, where I think pretty much nobody's going to perceive a social interaction between these two players. Okay? So we scanned people looking at video clips like these, and we looked for initially a contrast between the social interaction case, the top case, and the bottom case, the independent actions case. Okay, both of which involve 2 point light walkers of people doing activities. Okay, and what we saw was in 20 out of 22 subjects, an activation in a particular region of the superior temporal sulcus, shown here in 5 of the subjects. That's pretty good for a random crazy thing you try. Like usually you don't see something systematic like this. So that was sort of encouraging. And just to show you what that response profile looks like, this is now defining that region with some of the data and then measuring its magnitude of response in left-out data, so it's not circular. And what you see is a strong response to the interaction point light movies, much lower to the two independent activities and kind of intermediate for the mirrored one, which to me reflects my subjective sense of the strength of social interaction here. So that's pretty good and interesting, but we shouldn't believe it yet. It's easy to get things on a one shot. Maybe it's something weird about the point light movies, right? So next we said, okay, let's show some real videos. So we marched the lab up to Cambridge TV, where you can rent out a little video booth, and we filmed people in the lab doing simple little interactions like this. Aren't they cute? Betsy Don Blank and Terry Scott. Okay, so here's independent activities. Okay, so same basic contrast, but just with a video. Okay, all right. Okay, so what do we find? Same deal. Higher response to the interactions than the independent activities. Okay? It's a little less selective here. I think you can actually infer an interaction even for the independent ones. Anyway, we're now sticking all of these things on Mechanical Turk to get ratings of percepts of interactions, so it's not quite so hand-wavy. We'll see what that turns out. But basically, it's the same pattern. So then we say, well, okay, what about static information? Like even in a snapshot, you get a sense, okay, these people are interacting, okay, and these people aren't. Or maybe they're having a huff, I don't know. But it's not just proximity. These people aren't interacting, and those people are, okay? So they don't have to be looking at each other. So we scan people looking at stills of people interacting versus not. And there we pretty much don't get, I mean, it's significant with enough subjects, but it's minuscule, okay? I think this is related to the fact that right next door is that region that likes face motion. It doesn't like face stills, but I can't do much more with it than that. So it doesn't respond to any old interaction. I think those stills have plenty of information about interaction. But we have one more case where we asked whether a pure motion trajectory without body form is sufficient to engage that region. And so we returned to the classic from the, I think, 1940s, the Heider and Simmel study, where they showed all social interactions just from moving shapes. And so we made our own version of these. Here's an example of moving shapes that are socially interacting. It's a sweet little scenario. Oh, okay, heartwarming, right? Okay, in contrast, here is a video of two shapes that are moving in a principled way, but not socially interacting. These are not even animate. We have a third condition with animate shapes that I'm leaving out for simplicity. Okay, so now we can ask, does that same region respond to social interactions even of just simply moving shapes, totally different stimuli? Answer, yes, big time, okay? Even more selective in this case here. So where are we with this? Okay, and it's not just that social interactions are engaging. After all, we're social primates. We have all this social brain. Maybe we just like watching interactions. I think all that's true. But it's not the only reason that this region is engaged, because if you look in the fusiform face area, it does not respond differentially to any of these. It does not care at all, okay? Contra what lots of people have said for many years about how fusiform face area is really part of high-level semantic social processing. Oh, no, it is not. And you can see that with all of these contrasts. It just cares if there's a face there. It doesn't care about all this high-level stuff. So it looks like this region is doing something like perceiving dynamic social interactions. Okay, that's a little squishy. We haven't really nailed it, but it's tantalizing. And so what I want to do next is a number of things, including that method that Koster, Hale, and Sachs used to look at the patterns to see what's represented there. I want to see if you can tell from the pattern something about the nature of the interactions that you're perceiving. This will be a very interesting case to look at in autism. Both to look with pattern analysis in typical subjects, so we can look not just at the presence of the region, but the information it represents, and then to look at both things in autism. And we haven't done that yet, but are gearing up to. Okay, so this whole kind of woolly last section here, the superior temporal sulcus has lots of interesting stuff. It's not one big multi-purpose piece of social apparatus. It has structure in there. There are different responses, some of them quite selective, but we haven't nailed it yet. The face region in there is not a face region. It's doing something more high level. It responds at least as strongly to voices. There's much more to be done here. I think this region is very much worth understanding better in typical subjects. I think it's a prime suspect for deficits in autism. In fact, we've already seen that with the much lower response to moving faces in that region. Okay, so to wrap up, where are we? I would argue that the basic components of the social brain are pretty much present in people with autism, except possibly for that STS face region that isn't a face region. And I think we need to understand that region much more in typical subjects. There's a few subtle differences, like the language regions are slightly larger, they're less lateralized, and I think we need to know what the size of language regions and their lateralization means in typical subjects to understand that better. In terms of the way these regions interact, those look very similar from that resting functional study that I showed you for the language system. We need to look more at that method with the other regions. And one topic that Rebecca and Ev and others and I are particularly interested in is looking at pragmatic functions. These are how in language, these are really more what the deficits are in language processing and autism. They're not, basic syntax or semantics. They're things like understanding non-literal meanings, right? Like, did you go jogging? It's raining, right? That means you have to incorporate a lot of world knowledge to figure out that it's raining means, no, I didn't go jogging, because who would want to go jogging in the rain or something like that, right? That kind of high-level language processing that requires knowing more about the background of the thoughts of the person you're speaking with, bringing in more world knowledge, all of that kind of stuff, I think is a ripe area for study with autism, and I think we can look at it with the interactions between these language and theory of mind regions. Data I didn't talk about, but will if you ask. What about structural connectivity in the brain in people with autism? There's a lot of talk about how the major fiber bundles are compromised in autism. I think it's almost all, well, entirely or almost entirely a big artifact. I'll tell you about that if you want to know. None of the patterns I've shown you today can be significantly seen in contrasts of typical males versus females, although there are very, very weak non-significant trends for some of them in the same direction. I think one of the most exciting things to do with the future is to not just ask these basic first-order questions of, is the basic structure there, but to look at the information content of each of these regions. And I'll stop there. Thank you.